Matthew Kibble, who's here with us in the room, and this is very fortunate to have you here first in person. And Matthew Kibble will be speaking about, I cannot read it.
3D bioprinting.
Oh, 3D bioprinting. It's a very interesting topic. Matthew, the floor is yours.
Great, thanks. Do I change the slides with this?
Do you want to see on the screen? See this? No, I mean, do you want to be on the screen? Okay.
Well, if you move a little bit closer to the center, and you will be right in front of the camera.
It's pointing here, I think. So that's good.
¿Nos están escuchando?
Sí, sí se escucha.
¿Me confirman si nos están escuchando, por favor?
Sí se escucha.
¿Alguien me puede confirmar si nos están escuchando? Sí se escucha, ok. Gracias.
Okay, hello, my name is Matthew, and I'd like to start off by thanking the organizers for inviting me all the way to Mexico. And, your hospitality has been more than I could have anticipated. I'd also like to thank you for the opportunity to speak in person. The coronavirus pandemic has severely limited the opportunities for early career researchers to deliver to any audience, let alone an international one. So I hope that this is as exciting for you as it is for me. So a little about the university I come from, for those who may not be familiar. Sorry, do I? No. So the university is relatively young by UK standards. It's less than 200 years old. And in its current form is a mixture of several universities founded in the Victorian and more modern eras. And it's now one of the largest universities in the United Kingdom, with over 40,000 students, including, I'm told, 80 Mexicans, as well as 12,000 staff. So it's a very large institute, and benefits from being in the north of England, which over the past 10 years has seen increased government funding, increased movement of educational, political, financial capital in a bid to spread influence away from London, which has traditionally been the main centre of education.
I will change it for you.
Okay, thanks.
Maybe it is off. Maybe not.
And one of the major parts of this investment in Manchester is the Henry Royce Institute, which is a collaboration between several of the significant research institutes across the United Kingdom, including Imperial College, who we heard from this morning, and also a number of regional institutes in the north, including Manchester. The hub building is in Manchester and was opened last year, and brings together a series of research areas, for example, 2D materials, such as graphene, and atoms to devices, and biomedical materials, which is my own research area. And so whilst I consider myself a cell biologist, I am in fact a trained physicist originally. My undergraduate was at Imperial College. And I think that the transition of physicists and other technical sciences into biology is a really interesting growing area that allows interdisciplinary research in the areas where maybe one group alone does not have all of the research requirements to allow such complex projects to exist. So my own focus is on 3D bioprinting, which is the use of 3D printing-like machines to deliver biomaterials and cells together to model tissues, organ-like structures, and models of disease. And as part of that, we use many different biomaterials with the collaboration of biochemists, biomaterials engineers, medical doctors, and of course, physicists and those interested in fluid dynamics. In addition to this, other core specialties that I use in my own research particularly relate to bioimaging and the imaging of cells and the characterization of very soft materials. And so my project focuses on back pain, and I will use the intervertebral discs in the spine as the case study for 3D bioprinting. But this technology has been applied to many different tissue systems, for example, skin surfaces, the interface between bone and cartilage, and even areas as unusual as the blood-brain interface to model Alzheimer's, dementia, and other degenerative diseases. Why back pain? Well, in the UK and around the developed world, approximately 80% of all people will develop back pain at some point in their life. It's a major drain on national healthcare systems. In America, it's estimated to have a socioeconomic cost of over $100 billion per year. In the UK, it's $15 billion. Also in Mexico, there are some papers recently looking at the prevalence of back pain. 7% of Mexicans reported back pain within the last week, 69% over the course of their lifetime previously. And the lost productivity, the lost work days, the cost of treatment, which is either painkillers or invasive surgeries, is estimated at around $2,000 per treatment case. It's actually the 8th largest drain on the Mexican National Health Service today. And so the intervertebral discs are central to this medical problem. And in fact, they degrade more frequently than most other tissues in the human body, and are to blame in 40% of all cases. So it's a major cost that justifies a lot of research funds from very many areas, of which 3D bioprinting is one of the latest. So if we look at the intervertebral disc, I appreciate that there aren't many biologists in the room, so I'll keep it simple. But it's a fibrous cartilage-like structure between the bones in the spine. You have 23 of them, and they're made of a series of proteins, including collagen, many different types, and water, an awful lot of water, and proteins which are designed to keep water trapped. And it's a highly engineered structure. You see on the right of the diagram, we have a central gelatinous material known as the nucleus pulposus, and then outside a very fibrous, organized series of rings, about 25 of them, which together allow the discs to absorb compressive, tensile, and shear stresses, which, of course, with 3D motion throughout the day and the night, the body is constantly exposed to. They're very soft materials. So if we think about the elastic modulus of these, we're talking under 1 kilopascal for the nucleus pulposus in its healthy state, whereas as the tissue ages and degenerates as a result of genetics, mechanical wear and tear, and changes in cell biology, which will occur naturally, they become stiffer and they change their composition as well, which is why multiple disease models of development, health, age, and degeneration are all required in order to understand the processes that drive the changes to this organ. And so the intervertebral discs are 99% extracellular matrix, only 1% cell. And the extracellular matrix is the fundamental fixture in all living organisms. It's a series of proteins which are produced by the cells, which provide the support. You'll have heard of many of them from cosmetic adverts: hyaluronic acid, collagen, and also some non-fibrous components, including minerals, enzymes, water. Collectively, they build this very organized structure that feeds biochemical and mechanical signals through the cell membrane into the cells, which transmits the information about their environment to the DNA, which in turn will produce more matrix in response to the environment the cells are living in, healthy or otherwise. And so this series of cues, the aim with a branch of tissue engineering is to build systems that incorporate different cues and can influence the cell activity, shifting it away from a more degenerative, unhealthy type behavior back to a more healthy, younger, original state. And that's the goal of regenerative medicine. So looking at the intervertebral disc more closely, a slightly technical biological diagram. On the left-hand side, you have a typically healthy intervertebral disc matrix. You see the nucleus pulposus in yellow and the annulus fibrosus ring structure in blue, with very organized structures which, with time, begin to break down, driven by inflammation, particularly, and a series of signals released by the cells, which cause a cascade of changes that cause the intervertebral discs to generate faster and faster, until the only option is often total removal of the intervertebral discs. And so preventing this and encouraging regeneration of these discs and avoiding this surgery, either through preventative measures or ultimately through the transplanting of healthy tissue, is a real challenge for the 21st century, and one that has enormous potential for medical services around the world. So tissue engineering in its basic form, I know we have some bioengineers in the audience now, is the using of cells, for example, adult healthy cells, or maybe stem cells, which are cells which have the capability of turning into any type of cell. The most famous is the embryo, of course, but since 2004, it has been possible to take adult cells and reverse them back to their stem cell state, which removes the need to use embryos as a source of stem cells. These, in combination with biomaterials and scaffolds, for example, polymeric materials like PLA, polycaprolactone, and many other materials which you see in medical settings, such as titanium for implants, combined with additional factors and signals which encourage cells to make particular proteins. This combination are the building blocks of engineering tissues. And when you seek to do that and to engineer tissues, you effectively have two options, the top-down or the bottom-up approach. And so if we take the top-down approach, typically this would involve creating a scaffold, like, for example, using electrospinning, and then taking some cells from a tissue sample and allowing them to seed and perfuse into your scaffold. The idea in the long term is the cells adhere to the scaffold, they begin to produce their own matrix, and the scaffold has been chosen so that it degrades over time, leaving the cellular material that has been produced in its place. So material selection, obviously very important in this particular scenario. The other option is to effectively fabricate this yourself by using 3D bioprinting. So you were to take your cells and suspend them in a printable substance, which we call a bioink, and to design your tissue in advance and assemble the building blocks, the cells, the biomolecules, and the regulatory factors, so that you have instantly something that resembles a tissue already, and you then leave the tissue to do its own work in a bioreactor or incubator. this is the top-down approach is the more established route of the two, but there are some limitations. So if you're building a porous scaffold, waiting for your cells to move through it, you're obviously limited by the amount of diffusion that might take place into the center of the scaffold. You are also not able to encourage different cell types to partition themselves into different zones, for example, without creating discontinuous structures that you put together. It's quite labor intensive. And so automation is a longer term part of this idea as well, being that you remove the human element of assembling different parts together. Instead, you design it using principles similar to 3D printing. And as I've alluded to, the biomaterial selection is a really important part of this process. And there are many papers which have analyzed the pros and cons of the hundreds of biomaterials that are now available. I'll give you a selection here, and some of the things we have to think about. Given that we are working with cells, they have to survive. Like biocompatibility and longer-term rejection of the body is a key design constraint, as is in the case of 3D printing, whether you can put this material through a nozzle somewhere close to 37 degrees, because cells exposed to high or low temperatures, again, will die. Printing puts a considerable series of design constraints, which aren't at play for the design of acellular structures, such as titanium implants. Really, what you're seeking to do, because of the cellular components, is create an environment which encourages cellular response, but is not only biologically, but also physically and structurally relevant as well. So the choice of a collagen-based biomaterial, for example, is ultimately very inappropriate for many tissue types, because you're not able to generate very stiff constructs. If we take a look at different tissues, measured using compression or tensile testing, for example, and just sticking with Young's modulus here as a simple way of judging different tissue constraints. We start with fat tissue. We're talking about 1 kilopascal. Brain tissue, also very, very soft. If you dropped a brain on the floor, it would explode. Kidney, heart, and then much, much tougher structures such as collagen and such as cartilage and bone. Even these are nowhere near as stiff as the glass or tissue plastic you might be growing your cells on in standard cell biology environments. If you're growing cells, of course, another consideration is dimensionality. If you grow cells on a 2D layer, soft or stiff, the biomechanical forces the cells are experiencing are only in 2D. This is another justification for 3D engineered tissue models, because cells behave very differently when you give them 3D structures instead of 2D from experience. You also have to factor in local effects. So whilst a tissue's environment might be 1 kilopascal in a brain, for example, individual collagen fibers are immensely elastic and very stiff when measured in isolation using atomic force microscopy or nano indentation. We're talking gigapascal range, and these are some of the surface and topological effects the cells are also going to encounter, which will influence their response. So that's not to say designing the tissues is impossible, but it's definitely possible to overthink the design. And so in the biological side of things, it often becomes quite intuitive. I'm just seeking to show some of the complexity that goes into the design of the materials before the printing, which I mainly specialize in. So as I have already mentioned, the characterization of your materials is really critical. And this is a typical testing setup for human tissue, which is going to be very similar for hard metal, ceramic, polymeric structures. I'm sure some people in this room are familiar with this sort of setup. But with tissues, again, you have a series of protocols that you have to design. Tissue after surgery will begin to dry out. The fibers are already quite degraded, and they don't last very long. They begin to go off. And so on delivery from the hospital, you only have a small window of about a day before the tissue is not something you want to have in your laboratory, unless you freeze it. But of course, introducing freezing to a hydrated structure introduces crystals, disrupts the structure, and on thawing those structures, you have a different tissue. And so when thinking about your tissues and your tissue engineered equivalents, if you want to be sure that you're testing equivalent parameters, then you need to design a series of protocols that you can follow for both, involving hydration, involving maybe oxygen concentration, and also how the tissues have been treated in advance. Because whilst the nucleus pulposus is a very loose, unorganized, gel-like structure, the annulus fibrosus has a series of rings with an orientation. And if your piece of tissue is not aligned along the fiber, you're testing across it, then it doesn't have a lot of tensile stiffness. And so working with these samples is quite tricky. And this is where biomaterials expertise really does come useful. And there's an awful lot of very good work at Imperial, for example, looking at the biomechanics of intervertebral disc tissue. Now, 3D bioprinting, there are a series of different methods for bioprinting, one of which, extrusion, is quite similar in principle to the 3D plastic printing I think everybody here is familiar with. In this case, the extrusion is either done using air or a mechanical-like structure, such as a piston or a screw. But given that we're working with very soft and viscous materials such as hydrogels, which are 99% water with protein components, there are other methods as well, such as inkjet printing, which works in a similar way to an inkjet printer using piezoelectric acoustic oscillations or laser-based to project droplets onto a bed. And these droplets contain the cells and the biomaterials in the hydrogel. So these are an example of some of the new machines we have just had delivered to Manchester from a company called Selling. The one on the top is a 6 printhead extrusion bioprinter, although it also has some light curing capabilities, whereas the printer on the bottom left is purely a light-based printer. And so the bioinks that are used in this are photosensitive and are cured at different wavelengths of light, visible light as opposed to anything more harmful than ultraviolet light, because of course we're working with cells. And so the printability of your bioinks is a really key consideration. And these are just some of the outputs for a model bioink. Some of those variables I've already alluded to. If we take the example of alginate, for example, which is a material I work with, it's a polysaccharide derived from seaweed. And of course, the chains in the polysaccharide are of many different molecular weights, and this will influence the viscosity. And so you either have a broad range of molecular weights or a low molecular weight like version that you purified or purchased from a company, which will affect the viscosity and the shear rate and other components. Narrative to figure C, the shear switch. This is quite often the most indicative type of rheological study that we do on our bioprinted inks. Because we are looking at the G prime, the storage loss, and the G double prime, the loss modulus, which will determine the solid-like and liquid-like behaviors of your bioink, and the point at which your solid-like behavior begins to give way, and the bioink will begin to move through a nozzle and become printable. Temperature, as I mentioned already, a very important parameter. In the case of collagen, for example, the bioink will cross-link with temperature. And so if you hold your previously gel-like collagen at 37 degrees over the course of 20 minutes, it hardens. So what was printable is now not printable without a large amount of pressure, which will introduce shear stress, which may well kill the cells, and won't allow your printed structure to retain itself after printing, because you have already cross-linked it in advance. So it's quite a dynamic process, the choosing of the gels, and it's quite common to mix different biomaterials and bioinks together to incorporate multiple advantages and eliminate disadvantages of some of your systems. So for my research, I use a blended hydrogel of both alginate and collagen. The collagen will cross-link with temperature, but only after the alginate has been cross-linked with the addition of calcium chloride, which in large quantities is toxic to cells, but in small quantities results in a very rapid cross-linking of the alginate, meaning that you can print extremely soft, shear-thinning structures and then harden them later to give them a more realistic tissue environment for the cells to remodel. And this is data from my soon-to-be finished thesis, looking at the concentration of the hydrogel. As you increase the concentration, the linear region extends to the right, and the G-prime, the solid-like behavior, becomes larger. And the same with this cross-linker concentration. The more cross-linker you are adding, the harder your gel system is going to be after printing. And so through this, you're able to design a series of different regions with different cells in them, with different stiffnesses, which begins to look something like the intervertebral disc. Now, I know we have a lot of fluid dynamicists here, so I've tried my best to dig out my physics from 1 decade ago. So in the case of inkjet printing, there are a series of constraints on what can and cannot be printed, modeled through this quite simple looking equation, looking at the Reynolds number and various other properties relating to the ink viscosity, for example. And there's an enormous body of work looking at droplet mechanics, which biologists don't concern themselves with, but is certainly a very, very important part of bioink development, because not only do you need your cells to survive the printing process, you need whatever you've printed to retain its shape, and any large impact or collision of droplets or fusion of the lines that you've printed will impair the structure that you are trying to create. So there are many considerations that go into the actual printing, even though printing itself can seem like quite an intuitive process based on trial and error. The same applies for the extrusion versions. And one way that is quite often used in the development of new inks is to print a series of bridge-like lattices and to print your fluid across it and see at what point the line will retain its integrity and at what point this will fall apart. And such tests are useful for understanding whether the lines you print are going to maintain themselves or fall apart after you print it over the many weeks where you grow them in an incubator and the cells remodel them, because you need your original printed structure to keep its shape. Now, I mentioned that hydrogels are 99% water, and I'm working with a tissue of less than 1 kilopascal. So retaining the shape after printing is a really difficult challenge. You're effectively printing water, which won't keep its shape. And so one of the clever methods to overcome this particular design constraints is the use of suspended layer additive manufacturing. So I just go back to the slide I skipped. We take our standard bioprinting methodology, which is to isolate your cells from human tissue. For example, the 2 cell types in the 2 regions of the intervertebral disc. and grow them up until you have as many million cells as you need for your printed tissue, to then mix these into your hydrogels, your alginate collagen blended hydrogel that I use, to load into a printer cartridge and to print a disc-like structure. For it to not fall apart after printing, the solution with a dual series of technologies called SLAM and Fresh is to print into a supporting fluid. In this case, agarose, though you can use many other materials. These are shear thinning materials as well. But the fluid pressure that surrounds your print means that the print retains its shape, even though it is essentially a liquid, until you add your crosslinkers and heat it up, so that the collagen crosslinks, for example, and you end up with a structure which can be removed from the agarose and put into a bioreactor for growth. So this is a clever little way around the problem of working with very, very viscous solutions. And so this here is a series of tests that I used when working out the optimal combination of concentration of my hydrogels, the different supporting fluids that could be used, and also the settings of the printer, how fast it moves, what the pressure of the printer is when it extrudes. And you see, for example, very high pressure at the top. You have very thick lines which might even join together if you're working at small resolutions. And in the bottom right, with a less self-healing like material, you have perturbations in the lines that started to wiggle because the movement of the print head itself has introduced some vorticity. We're working in a 3D bath of liquid as well. And so printing too near the bottom of your bath will also introduce vortices. Printing too near the top, turbulence, if you like, ripples, or the introduction of bubbles. So there's a high degree of tolerance, but you do have to get this sort of thing that works out before just going on and printing materials with cells in, which is the fun bit. And so much like an STL or a CAD file, we design our tissue model, in this instance, a two-regioned, multi-layered disc shape, which I've used food coloring to show the two materials printed. You'll notice, of course, that there's no diffusion between them. They've retained their shape separate from one another as well, despite having different compositions and therefore densities. So this is a really effective system. So using multiple print heads with these different materials, it's possible to start building complex looking designed tissues, which you can then grow in an incubator and start looking at the biological outputs. Because this is one repeatable example, which has the same number of cells, the same biochemical composition, but you can then introduce factors to your growing time. For example, growing in acidic conditions, or low oxygen conditions, or conditions where you have induced inflammation through the addition of enzymes, to look at a cellular response. Everything up to now is entirely repeatable, assuming you've done your process the same up until this point. It's possible also to use the same cell type again and again by freezing stocks of your cells in liquid nitrogen, so that you can take them out at the same point that they were, and you have several copies of the same cell line that have maybe been processed chemically so that they no longer have age-like behavior. They've been frozen in time, and these are referred to as immortalized cells. And there are many immortalized cell lines available, including stem cell lines, which is very useful as a test before you start introducing human cells from real patients, each of whom has a different history, different genetic information, and cells in a different state. So here is the disc I have printed, only the outside. So this is to look at the resolution of the rings. Each ring is about 300 microns in thickness, which is large compared to, for example, inkjet printing, where you might be looking at microfluidic channels with cells that will migrate in different paths, for example, nerve regeneration studies. But my system does not need a really high degree of tolerance. So this SLAM method with a controlled printing process is adequate for my needs on the biological side. And you see here on the left the disc being printed in the supporting bath, after which I will remove to an incubator. And we haven't talked about cells. So cells respond to stiffness because they move and they latch on to the components of the extracellular matrix. So we have 3 examples here. Cells grown in 2D on a 1 kilopascal, 12 kilopascal, and glass. So a gigapascal substrate. And of course, you can see in the bottom one that the cells begin to stretch out. They take up more area. They begin to cluster together. And if you look very closely, you might not see this from the back, but they have a white skeleton inside made from actin, which is how the cells remodel themselves and start to move and communicate and change the matrix and sense what their environment is. So choosing a cell type that is responsive to stiffness, if that's what you need in your experimental model, which I do in the case of degeneration, because stiffness is one of the main drivers of disc degeneration. Selecting your cell types so they respond to your tissue environment is really important. It's also important to know that your printed structure, now it's growing after 1 week, 2 weeks, 3 weeks. It's retaining the design that you had for it. And so by using a fluorescent dye, which moves across the cell membranes, targets the cytoplasm and the components inside the cell, and stains them at different fluorescent wavelengths. You can then view them with fluorescent microscopes over the course of over a month, for example, to see whether the cells have begun migrating. And you see by day 35 on the right, we still have a very strong boundary between the cells. And there will be some areas where the cells are talking to one another, this NP cell and this AF cell. And it would be possible to take biopsies from these different areas of this gel and start to look at the biological outputs, for example, by staining them for different proteins. So immunostaining is a series of techniques to make available different binding sites so that you can view, for example, the locations of collagen type 2 or collagen type 4, or in this example, hyaluronic acid. So at the top we have human cartilage from the knees. The blue is a cell nucleus. The green is the hyaluronic acid the cells have produced. And this is in an adult human, so we all have knees like this. And the hyaluronic acid is very localized around each of the cells, or maybe cell clusters. Whereas in the case of intervertebral disc tissue, hyaluronic acid is an extremely important part of the hydrating mechanism that keeps the disc compressively useful. And so the hyaluronic acid is everywhere. Now, doing this in a bioprinted disc structure, we'll be able to look at the differences in the production as a result of collagen concentration, stiffness, the introduction of growth factors, and how this is different for different patients, for example, those with genetic disorders. You can also then start to look at how to encourage certain cell types, like stem cells, to start producing the right sort of matrix that turns them into healthy disc cells and restores healthy disc function. So this is one of the techniques that we use to understand the impact of different culture factors on the biological side. And another is PCR, which I think everybody has now heard of because of the coronavirus pandemic. The COVID virus has 3 genetic signals which are not present in humans. I think Omicron has 1 less than Delta did. But if these show up in this rainbow-like curve, this is a very accurate diagnostic. So to do a PCR test on a cell, you extract the RNA from the cell, use this to reconstruct DNA, and look at the amount of activity there was in a gene at that particular time. So you get a photographic snapshot of how much heat shock protein was being made on day 3, day 7, day 14, day 28, which will turn into protein production as an output. So these are a useful combination of techniques together to take a look at cellular activity and whether your model is biologically relevant and what the impacts of different cues are. Now, the hydrogels that we're talking about are several kilopascals at most, and I mentioned that the annulus fibrosus is in the order of 100 kilopascals with a series of very organized rings. And we have the organized rings, and we have a degree of cell alignment following the rings, but it's no match for an 100 kilopascal fibrous structure. And so as the technologies advance, we'll be looking to integrate things such as electrospinning of polylactic acid or PCL or other biocompatible fibers to create these lattice-like structures which can replicate the fibrous nature of any tissue, for example, and start to look at the importance of structural cues, not just physical, mechanical and chemical. What about porosity? What about fiber orientation? In the disc, the fibers orient differently in each ring. And of course, that's very difficult to replicate with any sort of printing technology. So this is something that potentially by the end of my project or a future project will look to be integrated into a bioprinted structure. And this sort of thing you can imagine is very useful for modeling bone, cartilage, and those porous shock-absorbing structures. So electrospinning is separate from bioprinting, but the technologies do converge at some points. And the Regenhu printer that I use, in addition to those selling printers I showed you, does have a melt electrospinning direct write print head next to it, allowing hybrid bioprinting. Now, as the field evolves, of course, this is the work of very many people, not only me and not only my university, but you can imagine the potential for tissue engineered systems with different cell types, instructive cues, and material characteristics to start looking at genetic disorders. For example, taking cells which have been gene edited with CRISPR-Cas9 or other editing technologies to start looking at development disorders in human development before the discs emerge, and tissues that create genetic disorders such as spina bifida, and also to use these models to investigate circadian rhythms and the compression that would take place over the course of a day and a night, and what impact this has on cellular activity and mechanical loading causing degeneration over time. So there are plenty of possibilities, including also the localized delivery of enzymes and chemical factors to encourage particular cellular activity. And one of the parallel research arms at the University of Manchester is to use graphene flakes and graphene oxide, this 2D material with a very high surface area. I mean, you can deliver very concentrated loads of growth factors to very precise locations. And so the development of bioinks containing graphene oxide is another research avenue that's been developing in Manchester over the past 5 years or so. So many possibilities. I'm very happy to take any questions you have on any of that. And just finally to mention, of course, you've brought me here to showcase my research and the research of Manchester. But of course, one of the reasons for me coming is to potentially plant the seed of new ideas. And so I'd like to highlight the 3 funds that I'm aware of: the British Council, the Newton Fund, and the World Academy of Sciences, all of whom have within their remits creating collaborations, workshops, exchange programs, and other activities looking at building research capacity between nations and institutes, such as the Pcr Institute here. Thank you very much.
Thank you very much for this very nice presentation. It's really amazing. Any questions? Several questions, I guess.
Yes. Considering that you're working with sometimes with viscous and not very elastic materials, how difficult it is to prove them, like how precise the machine needs to be, the movement, the displacement of the machine while printing.
So when I'm printing these, the blended alginate collagen structures, typically the extrusion pressure is about 0.02 megapascals, and the machine goes up to about 0.4 megapascals. So it's well within the tolerance of what is expected to be printing. I'm also using a very small diameter nozzle, 300 microns, but there are microns up to millimeters. Sorry, there are nozzles up to millimeters, which you could also use. If you notice in the lines I had printed, there was quite a lot of droplet diffusion on the surface. This is from the extrusion bioprinter, where ultra low viscosity materials are just dripping out without any pressure. So in my case, I use a microvalve based system which opens and shuts to avoid the dripping that would otherwise happen in just an open nozzle. So, yeah.
Do you have any questions?
Do we have any online questions? Because I'm here for days.
All right. Do you have any questions?
No, no.
Another question?
I have a lot of questions, but maybe outside.
Yes, of course, you can take it. So I have one question. Are there any clinical studies that says the viability of all these lab-made tissues?
Okay, so yes, there are. I'm aware of one that took place in Cambodia for skin grafts, like very severe wounds where there's almost no skin left after burn injuries to do any grafting. And then sheets of skin cells, like produced quickly, is better than nothing. That certainly took place a few years ago, maybe 2015 off the top of my head. The tissues that are often targeted are those more simple ones. Kidneys, for example, have 42 different cell types and enormous numbers of complicated structures like nephrons, which are bioprinted models in their own right, like bioprinted nephron systems, let alone kidney. But looking at, for example, nerve conduits, where motorcycle injuries have pulled apart the nerve, and then you're looking to produce some sort of structure which encourages the regrowth of neurons, maybe with a nerve guide, chemical factors, and a bioprinted connector. Those will approach clinical at some point. I mean, if you read the news, you'll think that bioprinted organs are already inside you. The reality is it's going to be several decades, I think, before that sort of technology is ready. But an array of model systems for drug discovery, for example, is a realistic possibility. And critical mass in the technology, it's still growing, but of course you have food printing and tissue engineering of meat-like structures as an entirely separate area, which I haven't even touched. And so the clinical translation is one of those areas, but I think it will take a while still until the cost of the technology has only just started coming down. The first inkjet printer was 2003. It's a very new technology. And a lot of the patents from 3D printing only expired 15 years ago, hence the explosion in commercial 3D printing technologies. I think there's some way to go. But companies like Selling are doing an enormous amount to democratize the technology, just creating cheaper, less highly engineered, but much more design-friendly versions of some of the more top-heavy specialist printers that are in big labs like Manchester's.
Well,
How expensive is the procedure?
Is the what, sorry?
Expensive.
So, several $100,000.
Just for a small disc, right?
Oh, sorry, the disc itself. No, the printer, the disc. A disc, like once you factor in the growing of the cells, which can cost different amounts depending on the cell type, the materials you've chosen. I think maybe a plate of discs for a triplicate experiment. I'm going to say $100 off the top of my head, which wasn't, in the context of science research, that part, relatively small for the creation of something like this. Sorry. Yeah. You just elaborate on that a little for me. Viscodissipation, you mean spreading after printing? Right, okay, yeah. So some of those formulas I referred to is used to govern what is printable. Coming out of the muzzle itself, you'll certainly get a little bit of curl using quite low pressures as a result. Certainly if I put the pressure up, you start to get a spray rather than a continuous stream. Remember, of course, my technique, it's already inside a fluid bath and the needle is in the bath as well. So it's limited, but there's a lot of molecular dynamic theory that's gone into that on the inkjet printing side in particular, to look at those windows of printability. Yes.
I guess you would be working in a very kind of laminar regime, right? So you push the fluid very slowly.
Yeah, so this is automated, of course, but yeah, just one of the big things for gel formulation is bubbles. You introduce bubbles, they stay in that, you print the bubbles, they burst, they might kill some of the cells. Like printing, generally, about 90% of your cells will survive, and then they grow and they repopulate. So printability, the viability is not really a problem, but certainly you don't want to expose them to conditions where they survive, but have altered function because of any oxidative stress, membrane damage. They might survive, but they might not be producing the healthy matrix that you expect them to. So there are tests for this, of course, looking at metabolism and existence of different proteins that are there immediately after printing. But they're generally fine with it. Cells can tolerate quite a lot, unless they're stem cells.
More questions? I have one last question, if you allow me. So how many cells survive the process? Because I'm guessing you have a death cell, a number of cells that destroy the process. And you end up with all sorts of biological garbage around the mixing with all these printing viral cells.
Yeah, viability is very high. Like I said, 90-something%. The general rule of thumb is that different cell types prefer different cell densities. The intervertebral disc cells, they don't like being in very cell-dense environments because the matrix is 99% of the disc. There aren't many cells in it. And in fact, for the nucleus pulposus, the cell density is 4,000 cells per millimeter cubed. It's not a lot. If you're looking to measure matrix production, you want an awful lot of cells. So I'll typically print with several million cells per centimeter cubed volume, just to make sure that after 1 week, 4 weeks, you have enough cell response to measure what you're looking at.
Well, if there are no more questions, I thank our guests very much. Tenemos una visita programada hoy, bueno, me parece. Entonces, les agradezco mucho su presencia a todos.